Okay, so when we're talking about different types of patterns of inheritance, codominance and incomplete dominance are going to be two examples of various different types of inheritance patterns. And so when you have a codominance occurring, that's when you're going to have more than one dominant allele contributing to a phenotype. And so the main example of codominance is going to be AB blood type, because blood type is determined by the type of sugars that you have on your red blood cells. And so the trait that codes for the A sugar and the trait that codes for the B sugar, they're both going to be dominant. It's not like A is dominant over B or B over A. And so they're going to work together in order to make the phenotype be AB type blood. And so codominance is when you have two of these dominant traits that are working together to contribute to a phenotype. All right, another type of inheritance pattern is called pleiotropy. And pleiotropy is when you have a mutation in one gene and it leads to all sorts of different bad negative side effects. Or not even negative side effects, but it leads to a whole bunch of different phenotypes associated with that one gene. And so a lot of these are associated with various different kinds of genetic diseases. And so a lot of times there'll be, a key protein or a key enzyme that's required, and there might be a mutation in that gene. So if there's a mutation in the gene, then that means your RNA is going to be messed up. But if your RNA is messed up, then your protein is going to be messed up. And that can often lead to disease. So a lot of these diseases are just caused by a mutation in one gene, which leads to just one misfunctioning protein. And so a lot of times when that can happen, you can end up with all sorts of different types of phenotypes. This example is looking at a disease called porphyria. And so you have a gene called porphyrin, and the one mutation in that one gene gives you this disease and is associated with all of these different types of phenotypes. So pleiotropy is when you have one gene that contributes to multiple different phenotypes. All right, another, not necessarily type of inheritance pattern, but something that can be observed in various different examples is something called epistasis. And so epistasis, what that means is there's actually two different genes that are determining what is being expressed or determining what genes are being turned on. But it's not like one is dominant over the other. It's usually like this gene over here will determine whether or not this gene over here is going to be turned on. So the effect or the phenotype is going to be relied upon by two different genes, or at least more than one different gene. And so one example of this is, we're going to go back to the blood sample. And so we've already said that blood codes for different sugars on the red blood cells. And so there's actually, in addition to the A or the B or the no sugars, there's also another gene that contributes to blood type, and it's called the H gene. And so when you have either a homozygous dominant or a heterozygous for this H gene, it's going to encode one of these molecule H proteins. And so whenever this molecule H is attached, that's going to associate with the type A sugar, and that's what's going to eventually going to produce the type A blood type. But if you have a homozygous recessive or a mutation in this H gene, then that means you're not going to have the molecule H, and that molecule H is required to attach those sugars to the blood. So even though this is coding for type A blood, if you don't have the molecule that helps attach it to the red blood cell, you're going to be a type O. Because type O, remember, is when you don't have any sugars. So in other words, you're having two genes and you're going to require both of them in order to see the phenotype. But usually one of them is going to take precedence over the other. So in this case, it doesn't matter that it's coding for type A, because you're missing that molecule that's needed to attach it to the red blood cell in the first place. Now, if this had been little I, type O genotype, if that was little I, and this was, homozygous dominant or heterozygous, this wouldn't matter because you're coding for type O, which means you don't need any sugars present, which means you don't need this molecule H. So it all kind of depends on the situation. But most of the times in epistasis, you have one gene that's dictating the expression of another gene. So in this case, this molecule H is dictating whether or not this blood type gets expressed. And the reason why is because this molecule is required to attach to the red blood cell. So if you don't have the molecule, then you're not going to have any sugars attached to the red blood cells, so you're going to be white. Not white, you're going to be type O. I don't know where that came from. All right. Another example of epistasis is labs. There's like chocolate labs and black labs and yellow labs. Well, there's two genes that determine what color a lab is going to be. There's the color code gene, like brown, black, so forth and so on. And then there's another gene that has to be, expressed in order for that coat color to be deposited. So it's this exact same example. And so this genotype, so if there's a gene that controls whether or not this pigment is being expressed, if you have a homozygous recessive of this gene, it doesn't matter what the color code is, whether it's brown or black or anything else, because if you get this homozygous dominant, that means by default the lab is going to have a yellow color. So we say that this gene right here, the coat color gene, is epistatic to this one, meaning this is the more important gene that's going to determine whether this one even comes into play. So in this blood cell example, we say that the gene that encodes what type of blood you're going to have is epistatic to this gene, to the H gene. So in other words, it's depending on this gene whether or not to determine whether or not it's going to be expressed or not. Does that kind of make sense? So you still have genes and you still have dominance taking place, but one of those genes can override a phenotype. In this case, the H gene, if it's absent or if it's mutated, then it doesn't matter what your genotype is going to code for your blood. You're going to end up being I.
So is the molecule H present for all types? So if that was going to be type B, would it be molecule B attaches to molecule H?
No, I think it's molecule H attaches all the sugars to the red blood cells. And since O, it's the only one that doesn't have any sugars, so if you don't have this molecule H, or you have a mutation in it, in other words, you're little h, or it's not functioning at all, then, it doesn't matter. You're going to end up being a type O, just because you physically don't have the ability to attach the sugars to the red blood cell.
That's going to be if you're homozygous recessive.
Yes, so, because in this case, this molecule H, it's dominant, right? So you can be big H, and have the molecule, or you can be heterozygous. So as long as you're heterozygous or homozygous dominant, you'll be fine, and that'll dictate whatever your genes encode for your blood. But if you're little h, or homozygous recessive, then that's automatically going to send you to a type O phenotype. Other questions? All right. Okay, so there's also several different types of inheritance patterns that we can observe through what are called pedigrees. And these pedigrees are basically like family trees that are related to inheritance of a certain trait or of a certain disease. And I'm sure you've seen these before. It's lots of squares and circles and lines, and then these people have kids, and then they meet somebody who has kids, and that person's infected. That's a pedigree. That's one way that we can trace the inheritance of a particular gene. And so there's several different types of inheritance patterns. We've already said that autosomes, those are going to be the chromosomes, those are everything except for the sex chromosomes, right? So those are 44 out of the 46 chromosomes we have. Well, if you have an autosomal pattern of inheritance, that means that this mutation that you're looking for is going to be in one of those autosomes. So it's not going to affect males versus females because everybody has a chromosome 22 from their mom and everybody has a chromosome 13 from their dad. So it doesn't matter, it's not going to affect one population over another because it's in all of your cells. And so these autosomal inheritance patterns are going to be mutations that occur in your autosomes. So in other words, in chromosomes 1 through 22. So we call that an autosomal inheritance pattern. Examples of this are, these kind of simplistic genetic diseases that we've talked about before. Huntington's disease, there's a mutation in one protein. It's that mutation occurs within one of the autosomes, so you end up getting the disease. Same thing with cystic fibrosis. Mutation in one gene that's somewhere on either chromosome 1 all the way up to chromosome 22. But if you have a mutation on your sex chromosome, then that's going to have a different influence on which people are going to be more affected than not. And so sex-linked inheritance is going to be when you're following a mutation that's going to be either on the X chromosome or on the Y chromosome. So basically autosomal is when you got mutations on chromosome 1 through 22, and sex-linked is when you have mutations on either of your sex chromosomes. So X's or Y's. And so when you have these sex-linked inheritance, you're going to notice that certain sexes are going to inherit these traits more than other sexes. And so one common example of this is going to be in color blindness. Color blindness is going to be a sex-linked trait, and color blindness is going to affect males more than it's going to affect females, simply because females, we have 2 X chromosomes, right? So if one of them has this mutated gene for color blindness, we have a backup chromosome. Whereas in males, they only get 1 chromosome, 1 X. So if they've got that mutant, then they automatically have color blindness. So that's why you see color blindness in males more than in females. But when you're looking, and this is actually colorblind showing, or pedigree, not pedigree, but Punnett square being shown right here. So the way that we write this out, because it's a sex-linked disorder, we write the chromosome. So a female's obviously going to be XX, and the male's obviously going to be XY. And so then we just put the traits. And so color blindness is going to be one of these recessively inherited diseases. So in other words, you need to be little c, in order to see the phenotype. So in this example, we have a mother who's heterozygous. So she's got 1 big C and 1 little c. So she's going to be normal because she's got the normal copy of the, colorblind gene. And so she's going to be a carrier, but she's not going to be affected by it. And she hooks up with a guy who has normal vision. So if he has normal vision, he's going to have a big copy of the C on his X chromosome. But this trait, this gene, it's only found on the X chromosome. It's not found on the Y chromosome. So whatever X he received from either his mom or his dad is going to determine whether or not he's colorblind or not. In this case, the father has normal vision, and the mother is just a carrier or a heterozygote for the colorblind trait. And so you do the Punnett square just like you would anything else. And so big C and big C, that's going to be, big C, that's going to be normal vision, no color blindness. Same thing with this example. If you take the X chromosome with the little c and cross it to the big C, this is going to be a heterozygote. This will also be a carrier, but not going to be affected. Same thing with this son over here. He's going to inherit a normal Y chromosome from his father and a normal X chromosome from his mother, so he's going to be normal. But then there's a 25% chance that this little c will come and interact with this normal Y. So now this son has a little c, and a y, which that little c is telling you that you're going to be colorblind. And so the reason why it affects males more is because if females have one bad copy, they're still okay because they've got this as a backup, right? So that's why females are not going to be as affected by color blindness as males. Now there is a scenario where you can have females that are colorblind. If you took a male that was colorblind and say, you know, marries a female that's heterozygous, then you're going to have the possibility. This one would be heterozygous, so that one would be normal. This one would be normal. This guy would be normal. But
Yeah.
no, that's a big C. That's a little c. So he'd still be a heterozygous. But then this, wait, okay, so that's another male. So, all right, the only way this can happen is if
Choose color.
colorblind marries colorblind, then you're definitely going to end up with all colorblind.
You could do it with, yeah, you were.
Well, if this is little c and that's little c, and that's little c, there's going to be all little c's. There's no big
Yeah,
c's.
she's
She's
The woman, if the
heterozygous.
woman's heterozygous, it can still
Yeah.
work, right?
She was
No,
heterozygous.
She's not wild type though. I know it's little c and big c is hard to distinguish, but she's colorblind and he's colorblind.
Yeah, but if, okay.
So she has to be.
So yeah, her X that she's contributing is going to be the colorblind X.
Yeah, but
And
if the top one was a big C.
if it was a big C, but it's not a big C.
But if it was.
If it was a big
The
C,
bottom left-hand corner would still make a colorblind child. If the top XC was a big C, the bottom left-hand corner would still be a colorblind child.
Sure, you could
Which
be.
is what you were trying to say first time.
Colorblind daughter.
Yeah.
no, because it would.
If she's
That
a
would be
carrier.
If this is big C,
No, that's a
No, that's a
little.
little C.
you're killing me,
The
man.
mom, if the mom
If the
had a big C, little c, and the dad had a little c,
Use a different
Yes.
letter than
She's a
C.
carrier. She's
Use
not
like Z's and
colorblind.
T's
She's
or
a carrier.
something.
All right, I see what you're saying. All right, capital C, right? Capital C, yes. Little c, yes.
There we go. That's what you
And
were
then
trying
the
to
son-
point out the first time.
The son would be
Yes.
blind, colorblind too, the second son.
Yeah, this son would be, yeah, he would be colorblind too, little c.
So really much in a nutshell, in a nutshell,
Oh,
if the mother, if her exes are gonna be recessive, that's gonna pretty much mean the kid's
Yeah.
gonna be colorblind. That's what the mother
If the
wants.
dad's
Yeah,
colorblind.
and so there's not gonna be a lot, there's not gonna be any male linked diseases because if there's a disease on the Y chromosome, it's not gonna affect me or any other female that has two X's. So it's just because you receive when fertilization occurs, you know, you're getting one of those X's from your mom and you're getting either an X or a Y from your dad. So if you're a female and you happen to inherit that mutant colorblind allele, but you get a normal copy from your father, then you're fine. But if you're a male and you're only getting that one X from your mother and she happens to be a carrier, then you're automatically going to be affected because you only have one X chromosome. So what you get is what you're going to have to live with, basically. And so these sex-linked inherited disorders tend to affect males more than they affect females. And that's simply because females have a backup chromosome in case one of these other genes is mutated, whereas males don't. They only get one chromosome, so they're stuck with what they got. We okay with this? All right. Okay.
Oh,
Yes.
I'm sorry. What if the mom had two, C's or
Big C's or little C's?
yeah, big C's.
Two big C's, yeah? Okay.
And dad had a little.
Well, then, so here's a big C, and this is a little c. So this one would be heterozygous, this one would be heterozygous. Everybody would be normal. This guy would be normal, this guy would be normal.
But their kids could have a chance.
But they're kids, yeah, because there's a carrier. If this lady marries another heterozygote, then you're still going to have a 25% chance of, having the colorblind gene.
How would she have to, capital C. How would
This
she
is,
be
sorry, this is a
homozygous?
little c.
No, not that one, but the parent, the mom in the first place.
It's
Um,
not colored.
Because she can't get a capital C from her dad. So
who says?
it
Yes, she
would be
can.
a little C, would be,
If he's not colorblind.
He's not colorblind, yeah.
but
Parents before those parents.
I thought the guy's gonna be colorblind no matter what, because he has the C.
Only if he has the little c. The big C is the normal. That's the normal gene. So if you got the big C, you're good to go.
If it's a heterozygous.
Oh, so if the mom is not a carrier and the dad is not colorblind, that's how the two big C's.
Exactly. So if the mom's a carrier and the dad's a big C, then the children aren't going to be colorblind.
They have a chance,
They have, yeah, well, I mean,
25%.
well, if this is,
No, if it's a big.
what do we say, if that's a big C, if everything's a big C, then there's going to be no color blindness, right? If this is a little c, there's still going to be no color blindness because they're all going to be het. And in this case, they're getting the big C for mom and the big C for mom, so it doesn't matter. But if this was a little c,
Colorblind.
right,
She's a carrier.
then she's a carrier.
Then the son would be it.
Exactly, exactly.
And the son would have 50% chance of being colorblind.
Sure. Yeah. Yeah.
Yes.
I have to draw it out. All right, so anyways. So these X-linked recessive disorders are, of course, going to be on the X chromosome. And so, again, because every male is going to inherit a sex chromosome from their mother, depending on which one they inherit, you know, that's going to determine whether or not the son or the males are going to show the phenotype. So the simple explanation is females have a backup chromosome and males don't. It's the simplest way to explain.